Alrighty, so we left off here speaking about smooth muscle, talking a little bit about its structure and how it differs from cardiac muscle and skeletal muscle, which we talked about as well last time. And so we're going to look at how smooth muscle looks under the microscope, right? We know that it doesn't have a sarcomere, so we're going to talk about how the filaments are arranged a little bit differently, and then how contractions are brought about in smooth muscle and how that differs from what we saw in skeletal muscle. Okay. So smooth muscle also contracts via the sliding filament mechanism. We talked about the sliding filament model and how actin and myosin are thought to be sliding past each other, but really in actuality, myosin is dragging actin. Okay, so we talked about that as being the sliding filament mechanism, and we do see that here in smooth muscle as well, but there are some crucial differences. First off, actin and myosin are longer in smooth muscle. They're also arranged obliquely in smooth muscle. So because we have this spindle-shaped cell, we've got this more oblique or slanted arrangement of the actin and myosin filaments. So this is the myosin filament here, this thick filament, and the longer blue filament is going to be actin or the thin filament. And we can see that they're not elongated or straight up and down the way they are in skeletal muscle, but they're slightly slanted. Another difference here is that because of this longer actin and myosin filament, we see much slower, much more sustained contractions. So myosin is going to drag along the entire length of actin, as we can see here, and that creates a longer, slower, more sustained contraction. As a result, smooth muscle contractions are going to be anywhere from 10 to 100 times longer than what we see in skeletal muscle. And that is because of the oblique arrangement and then the longer filaments, the longer actin and myosin filaments. So let's talk about what actually happens when a contraction is brought about, and then we'll look at the cellular mechanism that brings about the contraction. So this is our smooth muscle cell, right? This is our thick filament here, kind of studded in between the longer thin filaments. And when the muscle contracts, the entire thing will crumple down, synonymous with a ball of tissue. If you crumple down a ball of tissue, that's what will happen to the entire smooth muscle cell, as each of these thick and thin filaments, we can see here, slide all the way to the other end of each other. So this is going to give us a longer contraction when this entire smooth muscle cell kind of shrinks down or crumples down on itself. I also want to point out the dense body. So the dense bodies are synonymous with the Z disc. If you remember the Z disc or the Z lines in skeletal muscle, it's the point of attachment of the actin filaments or the thin filaments. The dense bodies can be compared to those Z discs, and they're basically anchoring the thick filaments that are located here, but also the thin filaments, which are sliding and kind of keeping them stable as they slide and this entire thing crumples down. Okay. So let's move on to look at the steps of smooth muscle excitation, contraction, coupling. So this is what happens in smooth muscle to regulate or turn on, turn off the contractions here. So we'll walk through these steps as we actually look at a schematic drawing on the next slide. So first off, we want to start out by appreciating some differences that we see here with smooth muscle that are not seen in skeletal muscle. First off, the source of calcium. Calcium is also the regulating agent for smooth muscle contraction, but the sources for calcium are a bit different. So the sources for calcium in skeletal muscle were only the sarcoplasmic reticulum. Here in smooth muscle, we see it's coming from the ER, but also it's coming from outside the cell as well. So the source for calcium for smooth muscle contractions also include extracellular calcium. So once that calcium comes into the cell via these voltage-gated calcium channels, so whenever that action potential arrives on this smooth muscle, we talked about the neuroeffector junction. Remember, smooth muscle is innervated by autonomic neurons, so postganglionic neurons. The synapse that relays that communication is going to be the neuroeffector junction. So we're going to have a varicosity, that's going to release some neurotransmitter, and then that is going to bring about an action potential on the adjacent tissue, which is this smooth muscle cell. When that happens, these voltage-gated calcium channels immediately open up. Calcium rushes into this smooth muscle cell, and then it's going to trigger the opening up of other voltage-gated calcium channels, which are on the endoplasmic reticulum. Remember, this is an organelle within the cell that is storing calcium. So we have calcium coming into the cell, and that begets more calcium that is being released from the ER. Now, when that happens, calcium is going to flood into the cytosol or the cytoplasm here and bind onto calmodulin. So calmodulin is a regulatory protein that is located within the cytosol of smooth muscle cells. And when we bind calcium to calmodulin, we create what's called the calcium calmodulin complex. Alrighty, so the calcium calmodulin complex is now an important regulatory agent that turns on another enzyme called myosin light chain kinase. And I'm just going to write this in here just so that we can write this in your notes, what this stands for. So myosin light chain kinase. Okay. And so this is an enzyme. We can tell by the last name of this enzyme, kinase, that this is going to phosphorylate some protein in this cell. Right? Remember, kinases are going to phosphorylate. Phosphatases are going to remove those phosphates or dephosphorylate. And so when we turn on this MLCK enzyme, that is going to bring about the phosphorylation of the oops. Okay, that is going to bring about the phosphorylation of, the myosin heads on the filaments that are located within the cytosol. And so we phosphorylate these myosin heads, and they are able to begin dragging across the actin or thin filaments, and that is how we contract smooth muscle. So the turning on of these myosin heads, and here they're calling them myosin light chain. That is synonymous with the myosin heads or the myosin filament. So don't get confused with this terminology, myosin light chain. So we phosphorylate these myosin heads, which brings about muscle contraction. Now, a part of the regulating of this process is that in the absence of calcium, MLCK is not turned on, but another enzyme is turned on. This is called a phosphatase. And again, I'm going to annotate this here so that you can make it clear in your notes. This is dephosphorylated by another enzyme called a phosphatase. So you can put the word phosphatase here as the enzyme that will reverse this action. So in the absence of calcium, phosphatases are turned on, MLCK is turned off, and we unphosphorylate the myosin heads or the myosin light chain, and that will cease or stop muscle contraction. So very similar to what we saw with with skeletal muscle, where calcium is regulating the contractions, turning them on by binding to troponin, turning them off by coming off of troponin and then that tropomyosin can cover up those binding sites. Calcium is also regulating smooth muscle contraction, but in a very different way. It's binding to a different regulatory protein. So here it's not troponin, but it's calmodulin. And it's turning on the contractions by turning on this enzyme, MLCK, and then turning it off in its absence and turning on phosphatases, which dephosphorylate or unphosphorylate the myosin heads and cease muscle contraction that way. All right, so that's what we can see happening here. In the absence of calcium, we unphosphorylate those myosin heads, no myosin ATPase activity, no cross-bridge activity. In the presence of the MLCK, we phosphorylate those myosin heads. We have myosin ATPase activity, which brings about cross-bridge cycling, and that results in smooth muscle contraction. Alrighty. Now, just like we saw, again, I keep comparing what's happening here to skeletal muscle so we can point out some important similarities and then some important differences as well. So what we saw in skeletal muscle is that for all of this to cease, right, in order for there to not be calcium here, we're going to have to actively pump calcium out of the cell. So we have these calcium pumps on the membrane of the endoplasmic reticulum, as well as on the cell membrane itself that's pumping calcium back into the ER and out of the cell in order to activate those phosphatases and cease the contraction. Alrighty, so relaxation of smooth muscle is brought about with phosphatases that remove the phosphate from the myosin heads. And remember, the phosphate is pretty much what's energizing the myosin head. Remember that ADP plus the inorganic phosphate. So if we remove that inorganic phosphate, there's no energy to bring about myosin ATPase activity. So that's what that phosphate is really doing. So when we phosphorylate it, we provide the energy. When we dephosphorylate it, we remove that ability for energy to be liberated to drive that process. And in order for that to be brought about, calcium has to be removed from the cytoplasm. That is brought about by calcium pumps, so calcium ATPase pumps that are on the ER membrane, on the cell membrane, pumping calcium back out of the cell and back into the ER. Another mechanism for getting rid of calcium in the cell is the calcium-sodium countertransport. So basically, calcium is being exchanged for sodium that's coming into the cell. And we're using countertransport, so the energy that's moving sodium passively into the cell can be used to pump calcium actively out of the cell as well. Now, as I mentioned earlier, the contractions that we see in smooth muscle are much longer, much slower, much more sustained than the types of contraction that we see in skeletal muscle. And that is partly because of the length of those filaments, as well as the oblique arrangement of those filaments as well.